The overall goal of the following experiment is to assess the induction of cellular Neu1 Sialidase activity in response to ligand binding to its specific receptor. This is achieved by first culturing cells on 12 mm circular glass slides in a 24-well tissue culture plate for 24 hours. As a second step, the live cells adhered to the glass slide are placed directly in contact with a previously prepared mixture of sialidase substrate and receptor ligand with or without specific inhibitors on a microscope slide. Next, the microscope slide is immediately placed under a fluorescent microscope in order to visualize and digitally capture the fluorescent images over time. Results are obtained that show changes in Neu1 Sialidase activity on the cell surface associated with the ligand-treated live cells based on the blue fluorescence intensity surrounding the cells under epifluorescence microscopy.
The main advantage of using this technique over existing methods like spectrophotometric analysis is that you use less reagents. You can also observe and capture images of sialidase activity in live cells.
This method can be used to answer key questions in the immunology field, such as looking at potentially novel ligand-receptor interactions and also looking at the effects of specific enzyme or protein inhibitors on receptor activation.
Demonstrating the procedure will be Dr. Amith and Dr. Preethi Jayanth, former PhD students from my laboratory who use this sialidase assay in their research studies.
Prior to resurrecting frozen macrophage cells, prepare culture medium using sterile filtered Delbecko's modified Eagles medium supplemented with 10%